Hello, biologists. Today we're going to talk about angiosperms, which are the plants that produce flowers and fruits. Here we have our phylogenetic tree of plants. Down at the bottom, we have our common ancestral green alga that gave rise to all the plants that we know and love today. Our outgroup is the charophytes, and then all the way up our phylogenetic tree with the most traits that are listed, including embryos, apical growth, vascular tissue, megaphylls, seeds, is our group, the angiosperms. They also have flowers. They use endosperm, they have fruit, and they have this process called double fertilization, which is weird, and we're going to talk about it. So angiosperms are incredibly diverse. There's more than 300,000 species. All of these angiosperms have flowers. The goal of a flower is to promote pollination. Now, this one is big and showy. This is a sunflower called Helianthus. This sunflower is looking like this because it wants to attract pollinators. Pollinators that see this flower will come to it and will visit it. They'll get their little body covered in pollen, and then they'll fly over to another flower and they'll deposit the pollen there, thus spreading the sperm to a different egg. Now, there's something called pollination syndromes, where if you look at the flower, you can tell who pollinates it. This is big, it's yellow, it's open during the day. Bees love sunflowers. There are certain sunflowers that are red and tubular shaped. Those would be pollinated by hummingbirds. If they're white and waxy, they're often beetle pollinated. If they're open at night, like cactuses, and they have lots of nectar and lots of pollen, they're often bat pollinated. So the flower fits the pollinator, including things like grasses. They are wind pollinated. And so they don't have petals at all because they're not trying to attract a pollinator. They're just using the wind. So the flower fits the pollinator, and that's pollination syndromes. Angiosperms not only have flowers, but they also produce fruit. So the fruit is the ripened ovary around the seed, and that ovary is important for distinguishing angiosperms from gymnosperms. The seed itself is the same as gymnosperms. So the seed is the endosperm, the embryo, and the seed coat, but now it's wrapped in this thing called a fruit. Angiosperms are heterosporous, so they have megaspores and microspores, and they're going to use this process called double fertilization. They also have more complex vascular tissue than gymnosperms do. Here we have the anatomy of a flower. A flower is really one of the key distinguishing features of angiosperms. We're going to start up here in this picture. We have the petals. These petals are made to attract pollinators. Collectively, you call all the petals the calyx, or sorry, the corolla. And then when this flower is a bud, it is protected by these sepals. These are usually photosynthetic. They're not trying to attract the pollinator. They're protective. And those are called the calyx. So corolla and calyx. Together, this whole thing is called the perianth. And the little stem that holds up a flower is called the peduncle. The stem that holds up a leaf is called a pedicel. If we look at the female parts of the flower, that's in this gray box right here, we're going to have the stigma. The stigma is the top of the female parts. That's where the pollen lands. Then we have the style. That's the neck connecting the stigma to the ovary. Here's the ovary. Inside the ovary, we have the ovules and the megasporangium. So inside this megasporangium, this is where you would find the ovules, and inside the ovules are the egg. So the ovules will contain the megaspores. Megaspores will be produced via meiosis, but then they're going to divide again via mitosis. And that's going to make a specialized set of cells, which we cannot see in this photo, but we will see in a new slide in a second. That's called the embryo sac. The embryo sac itself is the female gametophyte. So that will be encased inside of these structures, the ovules. The male parts of the flower are called the androecium. These are going to be all the parts in this gray box here. The male parts contain the anthers, which are the sacs that produce pollen. So here are the little pollen sacs. The pollen sacs are the microsporangia. The filament is what holds up the anthers. And inside the microsporangia are the microspores. The microspores are produced via meiosis, and they will develop into haploid pollen grains. To create pollen, we're going to look at this flower up here. It's got both the female parts, the gynecium and the androecium. We're looking at the top at the pollen sac, that's called the anther. Inside the pollen sac, you're going to have the microsporangium. The microsporangium contains the microsporocyte. The microsporocyte is going to do meiosis, so meiosis is occurring here at this step. We've gone from diploid to haploid. We've created 4 microspores. Now, each of these microspores is going to get a shell over it, and it's going to divide into 2 cells. We're going to have 1, the nuclei that will form the tube cell, and 2, the generative cell that will later do mitosis and turn into 2 sperm cells. This pollen itself is the male gametophyte. Now let's talk about forming the embryo sac. So we're still looking at this flower, but now we're in the gynoecium. We're looking at the female parts. If we zoom in the ovule, we have the integuments that will become the seed coat. We have the hole in the integuments to access the future egg. That's called the micropyle. So those are the same as gymnosperms, but now it's going to get different. So inside your megasporangium, you'll have the megasporocyte. The megasporocyte will have the megaspore. The megaspore will do meiosis. It will produce haploid. So we're now in the haploid color of blue, these haploid structures. The first haploid structure will be 4 megaspores, but 3 die out, just like last time. We're left with 1 surviving megaspore. And that surviving megaspore does something weird. It's going to do mitosis, and it's going to produce 8 nuclei, but only 7 cells. And collectively, these 8 nuclei and 7 cells are the embryo sac. So we have 3 antipodal cells up at the top. They are opposite the micropyle. Then we have this bigger area in the center. It is dikaryotic. It is called the central cell, and it has 2 nuclei. Those 2 nuclei are called polar nuclei. And then down at the base, right by the micropyle, we'll have 1 egg cell. And on either side, we have 2 nuclei, 2 cells that are called synergids. So 7 cells. The one in the center has 2 nuclei, so it's 8 nuclei. We're still inside the integument, and this is the whole thing is inside the ovule. Here's what it looks like in the microscope. So you can see the antipodal cells. You can see down here the egg and the synergids, and that's going to be where the micropyle is. Now, fertilization in angiosperms is more complex because 2 things are going to be fertilized, and we're going to create some triploid material. So it's really different. We will start down here in this flower structure. Here we have the stigma, the style, and the ovary. Inside the ovary, we'll have the ovules. So zooming in here, here's the ovary. Then inside the ovary was the ovule. The ovule contains the megasporangium. Inside the megasporangium, we'll have the megaspore mother cell. The megaspore mother cell will do meiosis. It'll produce 4 megaspores, but 3 will die, so it'll leave 1. That one existing megaspore will then go through mitosis. So we're going to zoom out over here, and that mitosis will allow it to produce 7 cells of 8 nuclei. Here are your antipodal cells. There's 3 of them. Here's the 2 polar nuclei in the central cell. The cell that's directly next to the micropyle is the egg, and on either side of the egg, those are the synergids. So 2 synergids. Now let's look at the anthers. Here is the anther. Inside the anther, you have the microsporangia. The microsporangia have the microspore mother cells. The microspore mother cells will do meiosis. Meiosis will produce 4 microspores. Each of those microspores will develop into a pollen. The pollen is going to have a nuclei that produces the tube cell, and then a generative cell that will divide into 2 sperm because we need 2 for double fertilization. Now we have pollination occurring. Pollination occurs when the pollen lands on the stigma. That's pollination. Now let's get into fertilization. To fertilize the egg, this pollen needs to get its sperm nuclei down to the egg cell. And that means it has to grow a pollen tube all the way down the style, all the way around the ovule, down to the micropyle. So this is the pollen tube growing down to the micropyle. And you can see there's 2 sperm nuclei. Those are from the generative cell that did mitosis. One of those sperm nuclei will fuse to the egg, creating a diploid zygote. The other one is going to fuse to the 2 polar nuclei in the central cell. So one goes to the egg, one goes past it to the polar nuclei, and it will fuse together, creating triploid endosperm. Wild, right? So those triploid cells are going to grow, and it's going to form the endosperm that surrounds the embryo. That's that little bit of light tan in there. The embryo is developed from the zygote, which was created by the fusion of the sperm and the egg. Here's the embryo. So that's the baby plant of the next generation. And then the whole thing is wrapped in a seed coat, which is made from integuments from the mother plant. Your little seed will land somewhere and it will germinate and it will grow into a whole new plant. So I've got some vocab there for you if you want to read through it on the PowerPoint slides. And now you get to practice it yourself. So go ahead and pause the video. This diagram has everything missing, so that's exciting. I like to start with the key. So don't forget the key tells you which is diploid and which is haploid. And then when you're adding vocab, start up here at the flower. All right, we'll go through the answers now. Up at the flower, I have my sporophyte. It is diploid. I have the male parts, which are inside the anther, and the female parts, which are inside the ovary. The anther has the microsporangium, which contains the microsporocytes. Those will do meiosis, and that is starting the blue phase, which is the haploid phase. When you do meiosis, you produce microspores that turn into the pollen. The pollen is the male gametophyte. It is haploid. Inside, there's a generative cell and a tube cell. Both of those are nuclei. Those nuclei are going to be used to create fertilization, but we're going to fertilize 2 things. In the female part, we have the ovary. The ovary contains the ovules. Inside the ovules, you have the megasporangiosperm mother cell who will do meiosis, and that will produce 1 megaspore. It does produce 4, but 3 die off. So that megaspore is then going to divide via mitosis, because I've already done meiosis, and now I'm haploid. It's going to divide 4 times, creating 8 nuclei, but it's only 7 cells. So here is the embryo sac. This is the female gametophyte. In the embryo sac, I have the hole that's the entrance to the integuments. That is going to tell me where my egg cell is. The egg is right next to the micropyle, which is the hole in the integuments. On either side of the egg are your synergids. Then you have a big cell in the center with 2 nuclei. That's the central cell, and it contains your polar nuclei. And up at the top, opposite the micropyle, you have 3 antiportal cells. Here we're going to have pollinization occur. In pollination, your pollen lands on the stigma. It grows a pollen tube down the style. That pollen tube reaches an ovary. Each one of these ovaries or ovules inside the ovary would need its own pollen. The pollen tube will grow right up to the micropyle. The generative nuclei divides by mitosis, producing 2 sperm cells. One of those sperm cells fertilizes the egg. and one of them binds to the two polar nuclei in the central cell. When the sperm cell fuses to the egg, we say that fertilization has occurred. Now this is a diploid zygote. The haploid phase is over. That second sperm nuclei fuse to the two polar cells in the central cell, creating the triploid endosperm. That endosperm will be the food source for your growing embryo. The zygote grows into the embryo. The embryo is the baby plant. The baby plant has a food source, and it's protected by the seed coat, which is made from the integument. That little baby plant will germinate, it'll grow into a whole new flower, and the process will start again. All right, now when we're thinking about angiosperms, we organize them in a couple of different ways. The first thing we do is say it's either a dicot or a monocot. Cot refers to cotyledons. A cotyledon is a primordial leaf. It's not a true leaf. Instead, it's just a bunch of stored nutrients that are left over from the endosperm, and they are used to feed the plant while it's growing. Here I have a pea plant, and I can see the cotyledons here. If there are two cotyledons, it's a dicot, and if there's one, it's a monocot. Here's the original stem, and this is the first true leaf of my pea plant. Here's the root, and it's branching into multiple different types of roots. This is a pumpkin seed, and in this pumpkin seed, or that could be a sunflower seed, this is the two cotyledons that are growing, and the seed coat is right on the top of it. Here's another pea plant. I can see the seed coat ripping open, and this is the cotyledon, not a true leaf, but all the storage material. And this is the first root that comes out. And then here is a little, maybe a artichoke or maybe a dandelion looking at the base of it. But these are true leaves, and then these two round flat things, those are cotyledons. So they're basically the nutrient storage, but not an actual leaf, because their goal is to store the nutrients and feed the embryo, not to do photosynthesis. All right, so monocots, they have a single cotyledon. If you look at their leaves, they're usually long and narrow, think grasses. They have parallel veins. If you were to cut them open and look at their vascular tissue inside of them, the vascular tissue would be all scattered. And their flower parts are often in threes. So this has 3 petals and 3 sepals. Monocots also do not produce wood. So there's no true wood. They're herbaceous. Even though they can get big and they can get hard, they're not technically wood because they don't have organized second growth. They're going to have multiple roots, but no major taproot. If you want to visualize a taproot, think about a carrot or a parsnip or a daikon root. Examples that we know and love: lilies, orchids, asparagus, palm trees, not technically wood, even though they're a tree, and grasses. Grasses are really important for us because a lot of what we consume for food are grasses. Think about rice, wheat, corn, bamboo, sugarcane. The world would be a very different place if we did not have these plants. So monocots are really important to humanity. Eudicots, which are now the word we use instead of dicots, they're used interchangeably. I learned a dicot, so I'm going to say dicot. Dicots can be woody or herbaceous. They can have fibrous roots or a taproot like a carrot. A carrot is a dicot. They're going to have 2 cotyledons. Think about a peanut. A peanut has those 2 halves inside of it. Those are cotyledons. They're going to be broadleafs. They're going to have veins that come out from a central point or not. They might be branched or not, but they're more complex and they're not parallel. And their vascular bundle, so this is the stem of something, is going to be in a ring. It's organized in a ring centrally instead of scattered around. The flower parts of dicots are usually in 4 and 5s, but they can be way more complex than that. Examples: sunflowers, roses, oaks, cherries, apples, maples, most vegetables, and fruit. So again, really important to us, but we sustain ourselves on angiosperms. Angiosperms are also known to create fruits, and fruits are ripened ovaries. So any fruit you've ever eaten, you're eating ovary tissue. Here is how we know it's ovary tissue. I have this flower here. It has the peduncle, which holds up the flower. It has the receptacle. It has ovary. Inside the ovary are the ovules. The ovules contain the eggs that will form the seeds. Then it has the style and the stigma. It also has the outer parts, these petals and these sepals. If we look at the fruit that this ovary turns into, we can see how this part changes and grows. So here's my fruit. This is a tomato. It still has its peduncle. It still has the sepals, but now the ovary has grown and become thick and fleshy. It becomes the fruit part of the tomato. Inside of it, we still have the ovules. These ovules here contain the seeds now. So the seeds of this tomato are inside of your ovary. We can see the style and the stigma on the opposite end. And all of this part that we eat, we call it the pericarp. The pericarp has 3 layers. It has the exocarp, the mesocarp, and the endocarp. I've got another picture showing those. But this full thing that we consume is the ovary. Here's another picture of an ovary. This is a drupe. It's a type of fleshy fruit. The skin of your peach is the exocarp. The middle part, like the bulk of what you eat in a peach, is the mesocarp. And in a drupe, the endocarp is hardened into a pit. That's one of the characteristics of this type of fruit. Inside of the endocarp is where you find the seed. So this is the seed, and everything else is all ovary tissue. Here's another type of fruit. This is an apple. We have the peduncle, which holds up the flower. Then we have the receptacle, which sits around the ovary. The ovary here contains the exocarp, mesocarp, and endocarp. Interior are these seeds. And if we look at the far end, we have the remnants of the stigma and the stamens and the sepals down here. It's the same for a pea plant too. The full body is the ovary, inside are the seeds. So what kind of fruits are there? If they're all ripened ovaries, how do we classify them? And we classify them based on the texture of their pericarps. So berries, they have an endocarp and a mesocarp that are fleshy with lots of seeds. For example, a tomato and a grape. Fun fact, blackberries and strawberries are not technically berries, and so they're in a different group. Drupes contain a hard pit. That's going to be your mangoes and your peaches. Palms have a central core containing multiple small seeds. That's going to be your apple and your pear. All of these are ovary tissue again. A Hesperidium is an endocarp separated into segments with a rind. That's going to be our citrus. Think about pulling apart an orange, each one of those segments. Then we have pepo. Pepos are modified berries, but they have a hard rind. For example, pumpkin and watermelon. So technically, pepos are types of berries. And then you have multiple fruits where the ovaries of nearby flowers fuse. That's going to be a pineapple. We also have dry fruits. What we think of as seeds and nuts are often fruits. So for example, we have archenes. They do not open, so they are indehiscent. You can split them open with your teeth, but they don't open on their own. For example, the sunflower seed and a strawberry. So each of those tiny dots on the strawberry, that's the fruit. And the red part, the fleshy part that you eat, that's the swollen receptacle. It's technically not a fruit. So strawberry is not a berry, and the fruits of a strawberry are what we think of as the strawberry seeds. Another type of dry fruit is a samara. It's a wingakene. So here's an example. The fruit is down here, and then it's got this wing projection that allows it to fly in the wind. And that hopefully will disperse it further from its parent plant so it can have a nice habitat to germinate in. We have Caryopsis. Caryopsis, you can't tell the difference between the seed and the fruit because they're fused. Examples are corn and rice. So corn and rice are fruit as well. Then we have legumes. Legumes split open along 2 seams. Think about beans or peanuts. You can split those 2 halves, and they split on their own when left on the plant. So they are dehiscent. They open up, not indehiscent like sunflower seeds. And lastly, we have nuts. Nuts have a stony pericarp with a single large seed, for example, acorns and hazelnuts. Things that we think of as nuts, like walnuts and almonds, are actually the seeds of fleshy fruits, usually drupes, and they're called trimas, but they're not technically nuts because they have a fleshy part outside of them that we don't eat. But pretty exciting. And I want to end with corn. So corn is incredible. It's way cooler than expected. And I'm going to explain why. Corn is a grass. Corn is wind pollinated. So here I have a female ovary structure of a corn, and I have all of this corn silk. Each corn silk is a stigma and a style that connects to an ovary. Because this is wind pollinated, every single one of the tips of these corn silk needs a pollen to land on it. Then the pollen has to grow a pollen tube all the way up down, all the way to the ovary, all the way to the ovule, into the ovule to get through the micropyle. And then it's going to double fertilize each of the kernels. So each kernel, and there's about 400 to 600 on average on a corn cob, has its own silk. So each corn kernel that you bite has its own double fertilization event. And it's waiting by chance for pollen to fly through the wind and land on the right place, and then germinate all the way down to do that double fertilization. The fact that corn gets pollinated so well that we have a cob full of pollinated, fertilized seeds is just incredible. Think about all the pollen that must get just blown out everywhere to try and make that happen. If you grow corn, you can try and help this process. It's called tasseling. You take the male anthers and you shake them around right above the corn silk. And so you tassel it to try and hand pollinate it, because otherwise you're waiting on this miracle of wind bringing the pollen to each of those corn silks, which is just, it's wild that it happens. So corn is so cool. I hope you have a great rest of your day. Email me anytime with questions, and we will talk soon. Bye.